How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor and your host. Joining us are two distinguished guests from Wasatch BioLabs, Chad Pollard, the CEO and co-founder, and Dr. Jonathon Hill, the VP of Science and Technology and co-founder. Together, they bring a wealth of expertise in genetics, genomics, bioinformatics, and cell biology, providing insights into the revolutionary advancements and potential impacts of targeted methylation sequencing. So thank you so much both for coming on. For those that aren't watching the video, you guys get the award so far this year for having the most professional podcast setup. So thank you so much for coming on the show. Like you've got awesome mics. You've got the sound treatment behind you. I'm impressed. You're showing me up a little bit.
Hey, well, don't get used to it. It's just the building we're in. But thanks for having us on.
Yeah, definitely. So let's jump into it. Tell us about DNA methylation. I want to kind of just give people that baseline for those that are like, I know of this, but I need a little bit of a refresher as we start getting into the nitty-gritty of it. So what is DNA methylation and why is it important? Because this is something that we've studied for a long time. We know about this, but I think in the last few years, epigenetics, methylation, it's become much more buzzwords and we're doing a lot more testing with it. So I don't know who wants to kick it off there. I'll let you guys kind of figure out who wants
let's
to
let the scientists let's let the scientists take that one.
So yeah,
I think that's a good idea. Dr. Hill, yes, take it away.
Yeah, I can talk about this all day long. So it's really cool because, we're used to thinking about DNA sequences and mutations that can cause disease or inherited traits, different phenotypes, things like that. But on top of our genome, we have these chemical modifications to the DNA that we call the epigenome, right? And these chemical modifications can be changed throughout a person's life due to different environmental stimuli in different disease states, and even naturally, just as different cells are doing their different jobs and becoming unique cells in the embryo, they get their own unique signature of these methylation patterns. And these signatures can allow us to study disease or identify cell types in ways that we never could before. And you mentioned we have studied this for quite a while, but really the technology in the last few years has really changed to enable a level of detail in these processes that we've never had before.
Yeah, I think that's very well said. And I love the analogy. I don't know who came up with it, but where it's like our genetics is kind of like a piano. And so the genes are like the keys, but epigenetics is actually like what keys are being played for like that note that is actually being played, because everything's there, but what's actually activated and what leads to, kind of the cell's decision on saying, okay, this is going to be active, this is not going to be active. So it's, epigenetics, epi is above, right? So it's kind of what is above the genetics.
Right. Yeah. I like to think of it what's layered on top. And you're exactly right. Every cell in our body has the same DNA, but it doesn't have the same chemical modifications, the same epigenetics for each of those cells. And so it gives us a whole new insight into how things are functioning.
Mm-hmm. And like on that note, you kind of brought it up, but why we're so interested in it is because those signals are so unique to cells, right? And so that's a lot of people think it's almost too simple when we talk about it, but we're taking those signatures that are unique to cell types and we use it for a lot of diagnostic purposes we can dive into it, but yeah, it's pretty amazing how stable those signals are and just how we can use them for those cell of origin type diagnostics. So.
And as we said, a lot of this testing is just new and how we can use this information to actually increase our knowledge of what is happening in a certain disease or in a person. So I think this area is just very rapidly growing. So when we're talking about targeted DNA methylation sequencing. I hear sequencing, I think we're reading the letters of the genome or a part of the genome, right? But this is a little bit different. Again, it's targeted DNA methylation sequencing. So I'm guessing, Dr. Hill, maybe you'll be the one to answer this. How does that actually work? I mean, how does it work? And then obviously I want to learn a little bit more about what conditions or indications, because sometimes this is part of the diagnostic odyssey, do people order for this? Like in what context? But probably helpful to understand, like, how does this even work? Because we're not necessarily looking at just the order of A, T, C, and G.
Right. And, this is the area where the technology has really grown in the last few years. Traditionally, what you would do in order to look at the methylation states is you would treat the DNA ahead of time with a chemical. And that chemical would actually convert all of the C's in your DNA into T's. Unless they were methylated, then they're protected from the chemical treatment. And so then you could sequence it and look for places where it was converted or not converted to measure it. That's difficult. It has a lot of problems with it. And it was hard to get good data out of that. Now with some of our third generation sequencers, such as the nanopore sequencer that we use, or PacBio is also able to do it, they can natively read whether that cytosine is methylated or not. And so it just comes out of the data. When you're sequencing, you're not just saying, is it ATCG? You're saying, is it ATC or methylated C or G? And that gives us that extra data. We like to think of it as a rich data set that's giving us everything we can know about that DNA, not just the sequence itself. So when would you use it? I think there's two major uses right now. The majority of it is still research type uses, right? Since we now have the tools to measure it, we can go into different disease states and look at methylation patterns in the DNA to see how they might be affected in the disease state, or to identify cell types that are affected by the disease and might be dying as a result of the disease, those kinds of things, and just learn about what's happening. Another layer of the mechanism. The other one that we're now starting to see as we've started to do these studies is unique methylation patterns can be indicative of a lot of diseases. And so we have a number of projects that are going on right now looking at different methylation patterns and how they correlate with different diseases.
Maybe if it's okay, I'll even share one of the examples. One of the big ones we focus on is applications in neurodegenerative diseases, right? These are diseases that progress for years and years before symptoms ever happen. I mean, that is the hallmark of the disease, is neurons dying too quickly and then leading to dementia and all these other horrible symptoms that go with it. So in that case, we utilize it as a way of detecting where cell-free DNA is coming from, and specifically, can we see if any of it's coming from specific cell types in the brain, right? If you're looking for Parkinson's-related dementia, then you should see dopaminergic-derived DNA in the blood, and you can pull that out with high accuracy using the unique methylation marks for dopaminergic neurons. Or ALS, you can look at spinal motor neurons, and even things like MS, you can look for ratios of Schwann cells or oligodendrocytes. And so there's some pretty amazing use cases where we utilize it to identify the presence of these dying neuron DNA cells, or DNA that's coming from these dying neurons, and it's super powerful and it's really stable. And so there's just some amazing use cases for it.
Wow. I didn't even think of it in that way. I want to make sure I'm understanding right, where if we're looking at like cell-free DNA or something, we can say, okay, if this is the DNA we're establishing and this is what we're focused on, we can actually tell what type of cell it came from based on what genes are turned on and off with this methylation. Am I getting it?
Yeah, 100%.
Okay.
And
yeah,
so then
that's very cool.
Yeah, and we can use that as a way to predict the level of neuron DNA in the blood and give diagnosis as saying, hey, you have elevated dopaminergic neuron cell death. If you don't slow that down, you're going to get Parkinson's disease. So some pretty cool methods using this methylation approach.
Yeah, and that ties in so well with last week's episode. We talked to Dr. Patrick Short, who we dived into the genetics of ALS. And so that really ties in so much with what you're saying and how much we're really learning in terms of the neurodegenerative space. And just if we can identify people that are very like pre-symptomatic, but we're starting to see those levels increase, like now is the time to potentially, I mean, this is not my area, but to be starting treatment because then if we can prevent symptoms or just delay them as far as we can, I can see this being really useful as like almost a biomarker, so that we could see, well, what level are you this month, this quarter, however often someone's tested that. And that's just one example in one area of healthcare. I think it's just so exciting to look at even where we've come from, because I would imagine a lot of the testing was with genetic conditions that are epigenetic in nature. I think of Prader-Willi, Angelman, that's probably where a lot of the testing started. Am I on the right track there?
Yeah, absolutely. And there's a lot of use cases. I mean, we're doing things for women's health, men's fertility, even in the pathogen space. Like we're looking at things for Lyme's disease. I mean, there's so many use cases where you can use this targeted approach to simplify diagnostics and make them way more actionable for physicians and cost effective as well. So
Wow. Yeah, it's so interesting. And in terms of the type of tech that you guys actually use in the lab, doing a little bit of research, I learned you guys use Oxford Nanopore Technologies, the Promethean platform. So, is there a reason why you guys use this platform? What went into the decision making of? I'm thinking you looked at accuracy, coverage, sensitivity, scalability, specificity, all of that. Why the Oxford platform?
yeah, so a lot of it came down to we were looking for a platform that we could do at scale, that we could like build a high throughput lab around, and Oxford Nanopore was perfect for that. On top of it, the science is really good. I mean, it's highly accurate. It's over 99.6% accurate for targeted or for identifying methylation. But when we're looking at, this targeted approach, we wanted to be able to multiplex, get highly, accurate data and process at a high level. So that's one of the things we're excited about. I mean, just to kind of put it in scale, on these flow cells, we can run like a minimum of 96 samples on it. We'd likely scale way more than that. One of our labs is only 2,000 square feet. I mean, it's a tiny lab, but using Oxford Nanopore, we can process roughly 300,000 samples in that lab at scale. And so you can just do a tremendous amount of processing in a much smaller space, and you can reduce costs quite a bit. So that's a lot of the stuff our team works on, and why we love nanopores. You can optimize and drive down costs tremendously compared to things like Illumina and PacBio. But on top of that, there's a lot of advantages in the technology as well. And I don't know, maybe you want to talk about Jonathon? Yeah.
Yeah, from the technology point of view, one thing that drew us to Nanopore was their ability to do the native methylation calling, right? We did not want to do that bisulfite chemical treatment. It can have variable results. It has a lot of baggage that comes with it. And so the fact that it can just do that natively really reduces the number of controls we have to run. It reduces sources of error that we might have in our experiments. And so with their unique technology, we were able to get all of that with the things that Chad was talking about.
Mm-hmm.
Yeah,
And
that's very, very exciting. Go ahead, Chad.
well, yeah, I was just going to point out from a data side as well, most high throughput labs tend to have multiple types of machines to provide data. I mean, in the case of methylation, for example, usually people doing these studies are looking for genetic factors as well as methylation or epigenetic insights. And so they have to do, usually right now, the standard is using Illumina's epic array and so they get methylation using that technology, and then they have a different sequencing approach, and then they combine that data. But with Nanopore, it all comes together, right? We sequence the native DNA. We can do all the standard genetic analysis like copy number variation that are super valuable in genetics. But we also provide not only DNA methylation, we provide hydroxymethylation, we can provide 6-MA. And so it just like takes all the data and puts it into one platform. And so it's just much more valuable for researchers as well. So.
Yeah, you have so much more content within that you're not simply sequencing and reporting that back, but you have all these other different areas that can, depending on the type of work that they're doing, could be really advantageous. And I imagine a lot of conversations went into what is important in terms of what you do want to offer. What was some of these factors when it came to designing panels and figuring out, well, what do people actually need? What do we want to offer? Like, in terms of figuring out, okay, what are we offering for people?
Yeah. Well, honestly, the only reason we started down the path of providing a targeted methylation panel was because we had clients come to us and they said, hey, I know you guys are based off Oxford Nanopore. We're using the EPIC array, and it's giving us a whole lot of data that we don't need. We're only interested in these 1,200 loci. Do you have any way to do a targeted approach so that we can get more sequencing depth at our loci and reduce our costs? And we said, well, we haven't done it before, but we're pretty confident we can figure that out. And I say this was a lot of work of Jonathon and his team. And they did an incredible job where they realized, yeah, we can not only target and sequence native DNA, but in terms of cost, we were able to take these clients and literally just move them from the Illumina platform to our platform, and we saved them 40%. So their margins immediately jumped 40% of what they were taking home. And that was kind of the genesis of it. And that's where a lot of the value is these companies that are looking at methylation or even looking at, targeted genes, and they just wanted a cheaper way of doing it that gave them all that data. So it wasn't necessarily something where we said, hey, let's build this and they will come, but it was more
the
they came
opposite.
to us and said, no one can do this, can you pull it off? And so we did, and it was interesting. Even Oxford Nanopore themselves said, we've had hundreds of groups approach us and we've been trying to provide a solution like this, and they themselves are interested in working with us to kind of help their partners as well. So it's been surprising and a little eye-opening, but there's a lot of people that are looking for this targeted approach to give them methylation signal. So,
and everybody wins. It sounds
yeah,
like in
100%.
terms of, just being able to develop the technology, your clients are happy. And even Oxford just being like, hey, this is great that our technology is being used in this way. And you mentioned something that there's a lot of like cost savings with it. Is that because it's on the nano level? So you don't have to use as much reagents. You probably don't need as much sample. Is that kind of part of the money saving aspects, or is it other things more like how much time is being used?
I'll take that one. So the main benefit is this idea that we can multiplex a lot of samples on a single platform. If you are doing a whole genome sequencing run where you want to look at the methylation data, it takes one flow cell per patient, right? But because of the targeted side of this, where we can pick out the regions of the DNA that actually are informative for whatever question we're asking, we can now scale that back. And we're typically targeting about 0.1% of the genome. And so now we can put 100, or if we had enough barcodes, 300 patients on a single flow cell and run them together. Obviously, we now just divided the cost of that sequencing among all those patients, right? Mm-hmm. And so that creates a huge cost savings. The flow cell itself is the most expensive component with any sequencing experiment.
Yeah, like traditionally, all these platforms that are out there, they're research platforms. I mean, they're designed to target as much of the genome as they can get. And once people find their signal and their biomarker, they just don't need all the data that's coming with those platforms. And so that's really all it is now they know their loci that they're looking for. And so we just hone in on that. We give them really high depth and we allow them to sequence tons of samples on one flow cell. So yeah, it's just allowing them to kind of hone in where they're looking at. And so that's how we reduce that cost.
Hence the targeted part of the name, that very much makes sense. If you only need so much information, why do you need to then sift through a huge mountain of data? Let's just go to exactly what, your clients are looking for. And to make it a little bit, because some of these concepts are a little bit abstract for people that maybe not are directly in wet labs and everything, or even dry labs. So what are some of the indications that people are sending you samples for? Like, what are they trying to learn? I know we talked about neurodegenerative at the top of the show. Are there other examples or other areas of health that people are looking more into to say, okay, this is what we're actually using the data for?
Yeah, there's a bunch. So I mentioned the neuro use case. Another popular one was fertility, and it was actually really similar where it was a men's health case where they were trying to identify if there actually was any sperm in the ejaculate. And so we use it as that for it has to do with surgeries and making it so doctors know, is it worth it to do surgery or not on a patient. there's some really cool aspects in women's health and identifying cancers through the menstrual blood, or even things like endometriosis by looking for endometrial based tissue. So just a lot of different use cases where people have come to us and said, hey, this would be really valuable. There's a company working with us for a cancer panel. So they're really interested because there's a lot of genetic factors that go into cancers, right? When we say, oh, you have pancreatic cancer, there's a lot of different genetic cancers within pancreatic cancer. And so they're really interested in having this panel that not only helps them identify cancer types, but gives them all the associated genetic information as well. So yeah, a lot of different use cases, a lot of people interested in it in ways that we hadn't even thought of until they came to us, but pretty cool.
Yeah, that's really interesting just to hear all the different aspects. Dr. Hill, did you have anything to add to that?
Yeah, one thing I want to add is I think we're at the very beginning here. Because it's a new technique, because we can do this for the first time ever, we're going to see a huge growth of different methods and different use cases that are going to come out of this. And like Chad was saying, we already see things that we didn't even think of, and there's going to be just more and more of those as we move forward. It's going to be fun to see.
Yeah, definitely exciting. And just that you're like, oh, thinking about it this way and this way. And then a client comes, well, I'm actually going to use it for this. And you're like, wow, like we're having an impact in this space, in this space.
Yeah.
So I think that's just really, really exciting. And for people that are listening and these researchers that are like, wow, we need to start looking at this aspect. So, maybe potential clients of yours, can you walk us through a little bit of the process of actually submitting samples and getting these results? Like, what do potential clients need to know in terms of kind of the nitty gritty? Like, this is what we can submit. This is the type of data we're getting out of this.
Yeah. So I'd say we have two approaches. So one is, researchers that are just looking to do one-off sample studies. It's pretty straightforward. You just go to our website, Wasatch BioLabs, and you see all the different services we offer. Anything from RNA sequencing to amplicon sequencing to our targeted methylation approach. And even with that targeted methylation approach, it's pretty straightforward. You just upload your bed file with the sites you're looking to target. It's a one-time fee, and then it's a per sample cost. So it's pretty straightforward for researchers looking to just submit samples. You ship it to us, we process it, and we give you all the raw data back to you to analyze. With entities and companies that are looking for a more custom approach, maybe already in the clinical space and looking to utilize the CLIA side of our lab, that it's a little more involved, where you work with our project managers. We get a really good feel for what you're trying to accomplish with this panel, and we help them kind of target the sites, get set up. And so it's a little more involved, but essentially the same process. Tell us where you're wanting to target, what it's used for, and then we can help generate the reports that you need for your clinical purposes.
Yeah, definitely. And how do you guys see this progressing in the future? I mean, especially Dr. Hill, you mentioned like this is very much the beginnings of looking at a lot of these, the methylation, epigenetics, and all of this. Where do you see it going? Do you see a lot of genetic testing in the future in the clinical space? Because we've been talking a lot of research side, but in the clinical space, do you see it where people have their, say, genome sequenced, or they have a panel or something along those lines, but it's starting to become standard where they're getting this methylation information along with it to help us analyze the genetics and how it's actually playing a role in disease progression? Or are we like way off from that?
Well, I think they have two complementary uses. Getting your genome sequenced can give you the idea of certain mutations that you might carry that might lead to a propensity for a certain disease, things like that. Really, what this epigenetics tells you is what is the state of the disease right now. And so, for example, going back to the neurodegeneration example, it can tell you you're starting to have neurons that are dying off, right? That's something you cannot get from the genetics because the genetics doesn't change over your lifetime. The epigenetics does. And so we can take a baseline, and then maybe a year you come back and now you have elevated levels of cell-free DNA from a certain neuron population. We say, okay, we need to monitor. The next year come back, it's a little higher, and we say, okay, it's time to start treatment, those kinds of things. And so I think, in my mind, the first big thing that's going to happen is we're going to have pre-symptomatic diagnosis of a lot of diseases. And as you were mentioning earlier, this is the key time where the treatments are more effective. They already have a couple approved drugs, many more in clinical trials that can slow the neurodegeneration, for example, but it cannot restore the neurons you've already lost. And so it's imperative that we catch that early enough that we can prolong that die-off as long as possible without any symptoms to give the person the best quality of life that they can have for as long as possible. That's the immediate kind of future. Beyond that, I think we're going to see future studies, maybe in the cancer space, et cetera, where epigenetic patterns will tell you how effective a certain cancer drug will be, or if a certain radiation treatment needs to be done. It can guide treatments based on the signatures of what's happening inside these cells. And it gives us an easily accessible, stable biomarker to do these things.
Yep.
Yeah. Sorry, Chad, did you want to say something?
And I was just gonna say, I'm the visionary, right? I'm the CEO that is biased, but I 100% think that it's already almost to that point where physicians and researchers are always wanting epigenetic data with their sequencing data. So I think it's the way diagnostics are going, and I think it's going to change the way diagnostics are done for sure to have both the epigenetic and the genetic side.
Yeah, I think prevention is always going to be better than treatment, right? In any area that we're talking about. And what's interesting about this is kind of two things I think about is like, as Dr. Hill was mentioning, with the precision medicine approach of saying, okay, this is this particular person's genetic changes, but then also the epigenetic changes and how that, evolves over time, and creating a very tailored approach for that prevention treatment, whatever stage they're at. But then also I think about from a business model, Chad, you were saying that, you're a little biased. Aren't we all that are here talking about different things, right? I think everything's cool in genetics. But I think it's so interesting from a business model that, if I go get my genome sequenced, that's pretty set. I mean, there are little caveats to that. Is the sequencing going to get better, more, accurate over time? Sure. But in general, all right, I have that, right? Mm-hmm. But when it comes to epigenetic changes, if I do something, today in 2024, I would probably want to look at that again a year later so often. So you're also looking at having a type of testing that is ongoing. And from a business model, I got to think that's very good in terms of setting yourself up for having these relationships with clients that are going to keep coming back because they need another round of the data as opposed to a one and done approach. So I think that's just, makes it very exciting on your side of what you guys have developed and being in the forefront of the future of this and being a trailblazer to say, hey, we're one of the leaders in this space. I'm imagining that you guys are just going to explode.
Well, yeah. And like, I think you hit it on the head. Like a lot of times in these tests, it's, no one likes to hear that their genetics are set in stone. There's not much they can do to change certain predispositions, right? But with epigenetic tests like this, you can get tested, and it almost allows you to take treatment into your own hands if you wanted to, right? You're able to get tested, see what baseline levels look like, and get on a treatment plan, and you come back and see, is it having any effect? It's why, like diets do so well, is because people actually have things to, like, they have physical appearance to compare it to and see if it's working. And this kind of gives that kind of same feel when you're looking at epigenetic signatures and they fall out of line, you see increased neurodegenerative disease, you get on a treatment, and then you can check again to see, am I improving, those signals. So
yeah. And what a great point on your side, Chad, of just that this is an area of genetics that we actually can change, like as opposed to we're born with the genetics we have, that's not going to change, but our epigenetics do change. And I think, something, if people want to dive into a really cool area of looking at twin studies and how identical twins over time, their epigenetics really changes. If you look at them at 6 years old versus 96, they're going to be rapidly different because they've lived different lives, right? Unless they're doing everything the same together. That's why twin studies are so interesting in genetics when they're identical. But I just always thought that was interesting. And there's that study of one of the NASA scientists that has a twin that stayed on Earth while he went up into space and
Yeah.
just looking at their changes. I don't know if you guys
Yeah.
have nerded out over that one. That was years ago. I got to find him. He's got to come on the show. If anyone has an intro for me, please. We have to have an astronaut on. So yeah, no, this is all just really cool stuff. And you guys are really just at the forefront of it. Anything else to mention as we close out?
I just want to reemphasize we're at the beginning and we're open to customers and people that have ideas on unique ways to use this. And we want to see where we can take this and how it can benefit people's lives, right? In a way that I think doesn't get mentioned enough. By lowering the cost, we've also made this more accessible to everybody. And people ask, why don't you just sequence the whole genome because you can charge more money? Well, we don't want to do that. We want to make sure that everybody can do these kinds of screening tests and be benefited from it.
100%. That, like, I would say that is our goal in all this stuff, is to truly change the way a lot of these diagnostics are performed. There's too many genetic tests that are just out of people's reach. And I think a lot of these approaches kind of change the way healthcare looks like, preventative healthcare for a lot of people. So yeah, we're pretty excited about that and excited to partner with people and help them accomplish that for their use cases. So
yeah, by making it scalable, you in turn made it accessible for people, and to have that in the prevention space, I mean, that is huge. So thank you both for coming on, nerding out with me for half an hour. I love doing these. It's really cool, especially in an area that I don't know too much about. I just love learning all of this. So thank you so much for educating me and the audience. Where can people find you to learn more and maybe start a partnership with you guys?
Just go to wasatchbiolabs.com. There's a lot of ways to contact us through there. And that's where all of our partnerships have come from this point. So yeah, go hit us up. Yeah.
And we'll have a link in the show notes for people if they are lazy and just want to click on the link. So feel free to check that out. Also available at dnatoday.com. Thank you guys so much.
Thank you.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.
We're all made of DNA. We're all made of the same chemical DNA. We're all made